So today's video is going to be gene regulation. Specifically, we're going to focus on prokaryotic gene regulation. We'll look at eukaryotic gene regulation next. But we know a little bit more information about prokaryotic gene regulation. So first of all, let's talk about why in the first place we would even regulate the genes. Okay? Why does it matter in particular which genes are being expressed? Why aren't they just constantly all of them being expressed? Okay, so we do need to regulate genes because it's a waste of resources if we're expressing these genes that we don't need the product. You know, for instance, let's say we're producing an enzyme that can digest starch, but you haven't eaten anything. There's no starch to digest. Well, that's a waste of products. It's a waste of resources. That's a waste of the raw materials. That's a waste of energy to turn that gene, to have that gene be on. So we regulate them to help maintain our resources. Okay, let's take a second and look at some brief overviews here real quick between prokaryotic and eukaryotic. Okay, so we're focusing mostly on prokaryotic today. Prokaryotic is what is considered more negative. And negative not in a bad sense. Okay, remember when we talked about positive and negative feedback? This is the same situation. So prokaryotic is more negative in the sense that the gene is active until things turn it off. So in the negative sense, we're turning things off or inhibiting them. And that can be done a couple of ways. You can see over here on this diagram on the side, for instance, you can have something that will interfere with the function of the enzyme in the pathway. Okay, so here's my pathway to form tryptophan there. So there can be something that interferes with the actual pathway. Okay, so it disrupts the enzymes of the actual pathway. You can also have something that inhibits the transcription of enzymes along the pathway. Both of those would turn off the gene, the active gene. So that's what's considered more negative. Eukaryotic is more of a positive approach. Okay, so eukaryotic is more of a positive approach in the sense that the gene is inactive until it gets turned on. So most of your eukaryotic genes are off until they are turned on. That's the positive part. Where a lot of our prokaryotic genes are on until they are turned off. That's the negative part. Okay? The eukaryotic genes are a little bit more difficult to regulate because there's so much more DNA that needs to be taken care of just because of the nature. The large amount of DNA in a eukaryotic cell compared to a prokaryotic cell just makes it a little bit more difficult to regulate it. Okay? So our eukaryotic genes, like I said, are usually off until they are turned on. Okay, so like I said, we're going to focus on our prokaryotic genes here. So let's first talk about what's called an operon. Okay, so an operon is going to be a group of genes, like writing letters on top of other letters. Okay, it's a group of genes that have a related function. So they're usually all transcribed together. For instance, if you look at the picture up here, okay, I've got 5 genes here. Okay, and so their products all work together. They have a related function. So they are all transcribed at the same time, so that all these products will be available that are needed at that particular time. Okay, there is a promoter region for the operon. Okay, the promoter region is going to be at the beginning. It's going to work for the entire operon. Okay, so the promoter, it's going to be a nucleotide sequence that is going to bind with the RNA polymerase. Okay, so you can see your RNA polymerase coming in here binding with the promoter. Again, it's a nucleotide sequence, so it's a particular sequence of nitrogen bases that they're looking for here. Okay, it's going to bind with the RNA polymerase, and it's going to start the process of transcription. Okay, it's going to start the process of taking that DNA and turning it into messenger RNA. Okay, the operator region is also located in the promoter. You can see the operator region there. Okay, and the operator, not the operon, but the operator, this is going to be an area of that promoter that can bind with what's called a repressor protein. And we'll talk a little bit more about a repressor in just a second. But the repressor would do exactly what its name says. It's going to actually inhibit transcription. Okay, so essentially the operator is a regulator spot there on that in that promoter region. Okay, that's going to start the transcription. So the operon with an N is the entire thing. Okay, all these genes that are related to one another are transcribed at the same time. Okay, it consists of the promoter, which is where the RNA polymerase is going to bind to start the transcription. Within that promoter is also an operator that is going to be where the repressor protein can inhibit, and then it's going to contain the actual what are called structural genes. Okay, and the structural genes are what's actually being transcribed. Okay, so it'll contain those actual structural genes as well. All of that combined are going to make up your operon. And many of your operons are controlled by what's called a regulatory gene. You can see that regulatory gene there. It's not necessarily part of the operon, but it's going to produce the repressor. Okay, so the regulatory gene is going to make the repressor. And like I said before, the repressor can bind at that operator region and inhibit the transcription of the rest of the structural genes. So the regulator gene it's what actually makes the repressor, which is going to be a protein. Okay, remember our genes code for proteins, so it's going to be a protein, and like I said, that's going to be able to inhibit the transcription of the operon by binding to the operator. So many of your repressors are going to have allosteric properties. We've talked about allosteric properties when we talked about enzymes. Okay, a lot of your repressors, proteins, are going to have a modulator associated with them. And that modulator is going to bind to the receptor at an allosteric site. Remember, your allosteric site was where something bound to an object. We talked about the enzymes. Bound to one region of the enzyme, and it changed the shape. A lot of times we talk about changing the active site. So it wasn't actually binding to the active site. It was binding to the enzyme, and that resulted in a change somewhere else. Okay, and our modulators will do that for our repressors. They can bind to the allosteric site and change the conformation of the receptor. For instance, if you look at your possibilities down here with these pictures, okay, when I look at possibility 1 here, okay, I can see that I've got my regulator protein. My modulator, in this case it's an inducer, okay, my modulator is going to change the shape there of that site. Okay, it's going to widen up that site when it binds. So that would be an allosteric property there. That repressor is changing shape. Okay, in possibility number 2, my co-repressor here binds to the site here. You'll see it's going to, if you look at that, the site on the opposite side, it's going to change the conformation. Remember, that's what our fancy word for basically changing the shape. Right? It's going to change the conformation of our protein, because remember, proteins are all about their shape. Okay? It's going to change the conformation of this repressor protein, and it can either activate or deactivate the repressor. These modulators are usually the product of the biochemical pathway. Let's say, for instance, in that very first picture we saw, they were trying to make tryptophan. The modulator would be the tryptophan. Okay? When there's enough tryptophan available, it's going to activate the repressor so that we stop the genes are turned off and it stops making tryptophan. Okay, so let's look at a couple of operon examples. So the first one we're going to look at is what's called the Lac Operon. And again, these are in prokaryotes. Okay, so we're focused on the Lac Operon, LAC for lactose. Okay, so the Lac Operon is what's called an inducible operon. Okay, so it's an inducible operon, which means that it is going to get turned on by a modulator that inhibits the repressor. Okay, so it is turned on by a modulator that's going to inhibit the repressor. So you can see in your picture here, okay, your repressor protein here, the repressor protein is this red one. Okay, remember that's produced by the regulator gene. And that repressor protein, okay, if you look at its shape here, can fit here in this operator area. But when the modulator binds to the repressor protein, like it's ready to do here, okay, so when the modulator binds to the repressive protein, it actually inactivates the repressor, which means that the genes